The Nova1 gene is fascinating. It plays a vital role in how our bodies function. Scientists recently discovered something amazing about this gene. Humans have a unique variant of Nova1. This variant is not found in other mammals, not even in our close relatives like chimpanzees. What makes this human variant even more interesting is its recent appearance. It likely evolved within the last few 100,000 years. This is a blink of an eye in evolutionary terms. This suggests that the human Nova1 variant might hold clues about what makes us uniquely human. The Nova1 gene is a master regulator. This means it influences the activity of many other genes. In fact, Nova1 affects over 90% of human genes during development. This is a testament to its far-reaching influence on our biology. Scientists are particularly interested in Nova1's role in the brain. It plays a crucial role in shaping the connections between neurons, the brain's communication pathways. This makes Nova1 a prime candidate for understanding how our brains evolved to support complex functions like language. To investigate the unique role of the human Nova1 gene, scientists turned to our small, furry friends, mice. Mice have a similar gene, but it differs slightly from the human version. By introducing the human Nova1 variant into mice, researchers could study its effects in a living organism. This type of experiment, where a gene from one species is introduced into another, is called a transgenic experiment. It allows scientists to study the function of specific genes in a controlled way. In this case, the goal was to observe how the human Nova1 gene would impact the development and behavior of mice. The results were intriguing. Mice with the human Nova1 gene exhibited changes in their vocalizations. This was a significant finding because it suggested that Nova1 might play a role in vocal communication, a trait closely linked to language in humans. Mice communicate with each other using a range of high-pitched squeaks. These squeaks are often too high for humans to hear without specialized equipment. However, scientists can record and analyze these ultrasonic vocalizations to understand mouse communication patterns. When researchers analyzed the squeaks of mice with the human Nova1 gene, they noticed distinct differences. The genetically modified mice produced higher frequency squeaks compared to their normal counterparts. This suggested that the human Nova1 gene was influencing the development of their vocal cords or the neural circuits controlling vocal production. Furthermore, adult male mice with the human gene exhibited more complex syllables during courtship. This finding hinted at a potential role for Nova1 in the more nuanced aspects of vocal communication, such as the production of complex sounds and patterns. The discovery that a single gene, Nova1, could alter the vocalizations of mice was remarkable. It opened up new avenues for understanding the evolution of human language. Scientists believe that the human Nova1 variant might have provided our ancestors with a selective advantage in vocal communication. This advantage could have led to the development of more complex language, which in turn facilitated social interaction, knowledge sharing, and ultimately the rise of human civilization. While this is still a hypothesis, the findings surrounding Nova1 provide a tantalizing glimpse into the genetic underpinnings of our unique ability to communicate. The research on Nova1 is far from over. Scientists are eager to unravel the precise mechanisms by which this gene influences brain development, vocalization, and potentially other aspects of human cognition. The journey into the depths of our genetic code continues, promising exciting discoveries that could illuminate the mysteries of human evolution and the origins of our extraordinary capacity for language.